Good evening, everyone. Hello, good evening, welcome. My name's Athene Donald, and it's my pleasant task to welcome you all tonight for this very special occasion. So, good to see you all. I now have to do the air hostess bit. So, fire exits there, and I guess at the back too. Please turn your mobile phones off because they can interfere with the recording, and this is being webcast and recorded, so it would be good not to have any noises off. So it's my pleasure to introduce Sarah-Jayne Blakemore tonight, the winner of the Royal Society's Rosalind Franklin Prize. Now, this prize is awarded annually for an outstanding contribution to any area of science, technology, engineering, or mathematics. But in addition to the splendour of the science, the recipient is also expected to spend some of the prize money on some kind of project around raising the profile of women in these subjects. And the choice of what the people who apply for this prize or are nominated for this prize do is up to them, but it does form part of the sort of prize judging process. And I think we will hear a bit more in the talk about what Sarah-Jayne herself is going to do. So Professor Blakemore is a Royal Society University Research Fellow. She's a Professor of Cognitive Neuroscience at UCL, where she's the leader of the Developmental Cognitive Neuroscience Group at the Institute of Cognitive Neuroscience. And her main emphasis in her research is on social cognition and decision-making in human adolescence, and that is, of course, what we will hear about tonight. She's very much involved in public engagement with science and gives many public lectures, and she also talks to schools. She's worked with the Select Committee for Education, and indeed, she and I sit together on a committee in the Royal Society which is looking at what our vision of education should be 20 years hence. And she's also been a scientific consultant on the BBC series The Human Mind. So she has a long and impressive history, as it were. And she has been awarded the Royal Society Rosalind Franklin Award for her scientific achievements. And again, we're going to hear more about this in her talk on the teenage brain. So Sarah-Jayne, over to you.
Thank you, Athene. It's a great honour to have been given this award, and it's a real pleasure to be invited here this evening to tell you a bit about our research on the teenage brain. So, adolescence. You can think of adolescence as broadly concomitant with the teenage years, but actually I prefer a different definition of adolescence. I prefer a definition of adolescence which takes into account individual differences in adolescent development. So the definition that I like is the period of life that starts with the biological hormonal changes at puberty and ends at the age at which an individual attains a stable, independent role in society. So you can see that adolescence can go on quite a long time. And in some countries, like in this one, it's perfectly acceptable for individuals to stay at home and be in full-time education right into their 20s and even their 30s. But that's not the case in other cultures. There's a big cultural variation in the concept of adolescence. So in some cultures, it's not acceptable to stay at home post-puberty. Children are expected to go out and become financially independent, get jobs, get married, have children as soon as they reach sexual maturity. And some people have used these big cultural differences in the concept of adolescence to argue that adolescence is a kind of recent Western phenomenon invented in the USA about 50 or 75 years ago. But actually there is some evidence that adolescence, this concept of adolescence and adolescent typical behaviours are more widespread, more culturally invariant, and can be seen in portrayals of adolescence throughout history. So one really good example of this is this quote by Shakespeare from The Winter's Tale. So here Shakespeare says, "I would there were no age between 10 and 3 and 20, or that youth would sleep out the rest, for there is nothing in the between but getting wenches with child, wronging the ancientry, stealing, fighting." So Shakespeare, almost 400 years ago, is describing this age group in a very similar way that we stereotypically describe this age group today, and the way they are portrayed and sometimes demonized in the media. Another example of a description that really nicely characterizes what it's like to be an adolescent is this letter which was sent to The Guardian last year. So this is a letter from a reader of The Guardian. She says, "There's nothing like teenage diaries for putting momentous historical events in perspective. This is my entry for the 20th of July 1969. I went to the art centre by myself in yellow cords and blouse. Ian was there, but he didn't speak to me. Got rhyme put in my handbag from someone who's apparently got a crush on me. It's Nicholas, I think. Ugh, man landed on moon." This, I think, really nicely illustrates what's important to a teenager. Not the fact that man landed on moon that day, but things like what clothes you wear, what music you like, your political moral beliefs, who you hang out with, how other people see you. Something that undergoes profound development during adolescence is your sense of self, and in particular, your sense of social self, how other people see you and how you behave in front of your peers. And there's a big, important drive in adolescence to become independent from one's parents and one's families, and to seek the approval of one's peers and move towards one's peers. So recently, adolescent typical behaviours have started to be studied in the lab, in many labs around the world. And one of those adolescent typical behaviours that we tend to associate with teenagers is risk-taking. So it is true that in certain circumstances, in certain contexts, teenagers will tend to take more risks than any other age group. It's very much dependent on the context and the kind of risk we're talking about, but there is some evidence of that. And recently, people like Larry Steinberg at the University of Temple in the USA have started to research risk-taking in teenagers in the lab. So in a typical experiment in Larry Steinberg's lab, he brings people into the lab and he gets them to play a driving video game, just like the one you can see here. So in this game, it's like a kind of video arcade game where you drive around a track, and he can measure the number of risks you take. So he can measure things like how many red lights you go through, how many times you speed, and how many crashes you have. And he tests different age people, either when they're on their own or when they have a couple of friends standing behind them. So these are data, what this shows are the number of crashes people have. In fact, it's very similar no matter what risk you look at. And what you can see here is the number of crashes that people have when they're on their own. The red bar is data from the adolescents, so these are 13 to 16-year-olds. The green bar are data from young adults, 17 to 24-year-olds. The white bar is from adults 25 and over. So you can see that when they're on their own, actually they take a really very similar number of risks or have a similar number of crashes. However, when they have a couple of friends standing behind them, that very significantly increases the number of risks adolescents take, and also increases the number of risks young adults take. And it doesn't have any effect on the number of risks that adults take. So this idea that what really matters is peer influence, whether there are friends with you at the time, that determines whether adolescents take risks. That is borne out by, if you look at data from, for example, driving insurance companies, you will see that the number of road traffic accidents is higher in young people than in older adults, but that if you look into that further and look at what context, what circumstances young people are having driving accidents, it's normally when they have a similar aged passenger in the car with them. And that really is the same kind of phenomenon that we're seeing here. So it seems that one really important factor in driving this risk-taking behaviour is the presence of peers. And so we're really interested in finding out why that is. What is it about peers that is so important to the adolescent? And one of the ways we've looked at that is to use what's called the Cyberball game. This is an online experimental social exclusion manipulation. So in this game, which was developed by Kip Williams and his colleagues in the US, you are a participant here, and you come into the lab. You don't have to do it in the lab, but in our case, we did it in the lab. You come into the lab and you're told, right, you're going to play a ball throwing game with other people over the internet, and the other people are represented by these characters here. In fact, there are no other people, it's all programmed by us. And we can then manipulate what happens during this game, and you can either be included in the ball game or not. So in this game, there'll be a ball, and you can throw it to either of the other two people, and they can either choose to throw it back to you or not, and you can see a kind of simulation of this here. So first of all, you get the ball, you choose who to throw it back to, and they could have thrown it back to you, but in this case, this is an exclusion condition. They're leaving you out of the game. And this is very sad. We know from data from Kip Williams' group that adults, when they're being excluded in this kind of game, they find this distressing. You can see significant drop in their mood and significant increases in their anxiety levels. So with Catherine Sebastian, who was a PhD student in my lab and now runs her own lab at Royal Holloway College here in London, we were interested in how adolescents would respond to being excluded in this kind of online ball throwing game. So if I just show you some data, we carried out this experiment with a group of young adolescents, a group of mid-adolescents, and a group of adults. I'm going to show you firstly data from the adults. So what you can see here is the overall mood, which is measured by a questionnaire. And these are data from the adults. What you can see here is that this is the data at baseline, so that's before the experiment started. And this is after inclusion, so after being included in the ball throwing game by those other two players, and after being excluded by them. And for adults, there's a significant drop in mood after being excluded in this ball game compared to after being included. For the young adolescents and the mid-adolescents, the data look very, very similar. The pattern is the same, except the drop in mood after being excluded in this game is significantly greater in both young adolescents, so they're 11 to 13-year-olds, and mid-adolescents, 14 to 16-year-olds, compared with the adults. So what this suggests is that adolescents are hypersensitive to being socially excluded. More recently, another of my PhD students, Kate Mills, and I have been thinking about what this means for adolescent behaviour, and we're starting to work with this framework to think about adolescent behaviour and adolescent decision making. So if you think about any kind of everyday life decision that you have to make, for example, we make decisions that have some nominal risk all the time. So for example, you might be deciding whether you're going to drive really fast. Shall I drive over the speed limit? When you're making these kinds of decisions, you weigh up various advantages and disadvantages. So in this case, you might think, well, yeah, it'd be really useful to drive really fast because I'll get to my meeting quicker. And you might even get a kick out of driving really fast down the motorway. On the other hand, there are also disadvantages which will come into the decision-making process. So you might crash, or you might be caught speeding by the police. Those kinds of factors will weigh into whether you decide to drive really fast or not. But there's another factor that also comes into play, and that's the social factor. We all behave differently when we're in groups of people compared with when we're on our own. That's true for adults and for children and for adolescents. Our performance on tasks is different when we think we're being observed by someone else. But what we're suggesting here is that this social factor weighs in particularly heavily for adolescents compared with any other age group. The importance of what friends think is heightened during the adolescent period. And if you think about adolescent typical behaviour and decision making, in this kind of framework, it sheds adolescent behaviour in a more rational light. So often adolescents get demonised for making bad decisions or taking crazy risks. But actually think about the kinds of risks that adolescents typically take, things like starting smoking or binge drinking or taking drugs. Imagine you have a 13-year-old girl and all her friends are smoking cigarettes. They offer her a cigarette. What's the bigger risk for her? To say, yes, I will just try that cigarette, knowing, as all 13-year-olds do, they're not stupid, that, that smoking has a measurable risk associated with it, or saying no and potentially being ostracized by that peer group, but potentially being excluded by her friends. I would say that there's no competition. The latter risk is much more serious for an adolescent, the risk of being socially excluded. So in fact, if you think about adolescent behaviour in that kind of, using that kind of framework, it seems like often the more rational choice. So what about the adolescent brain? Until about 15 years ago, we really had very little idea about what is going on in the developing human brain. That's because we had no way of looking inside the living human brain. All our information about how the brain develops until about 10 or 15 years ago came either from animal studies or from post-mortem human brain studies, which were very, very scarce. And until about 15 years ago, if you asked a neuroscientist, when does the human brain develop? They would probably have not had a clue and just had to guess. And I would say that the vast majority of neuroscientists would have said sometime in early or mid childhood, because we just had no idea about brain development continuing beyond then. In fact, it turns out that's absolutely not true at all, and brain development goes on for right throughout adolescence and for the first few decades of life, and that's what I'm going to talk about in the rest of this talk. So how do we know that? We know that because of advances in imaging technology. So particularly MRI, or magnetic resonance imaging, has been used by many different groups around the world to track changes in the brain, particularly in brain structure and also in brain function across the lifespan. So MRI, some of you may have had an MRI scan. It's essentially a very large magnet which you lie inside, and because different structures have different magnetic properties, you can measure the structure of the brain and how that develops across the lifespan. So this is a friend of mine's brain, the MRI scan of a friend of mine's brain. You can even see, probably from where you're sitting, the very detailed convoluted surface of the brain. That's the cortex. You can see her eyeballs. This gives very high resolution images of the structure, like a three-dimensional photograph of the living human brain. And if you want to measure things, you can measure grey matter volume. So grey matter is mostly found in the surface of the brain, the cortex. You can see that here. This is a slice like that through the brain. This is the front, this is the back. And grey matter contains many different types of tissue, including cell bodies, so neuronal cell bodies, and connections between cells. They're called synapses. You can also measure white matter. So white matter is found in the center of the brain, and it contains the long fibers or axons that connect up cells together. And these very big white matter tracts connect different regions of the cortex with each other. Using MRI, you can also measure brain activity using functional MRI. So this isn't really neuronal activity, it's a proxy of neuronal activity. What we're measuring when you see pictures like this, which you do often in newspapers and on the TV and in science papers, is not neuronal activity. It's essentially where blood is flowing to in the brain when you do a particular task. So we might get people to lie inside the scanner and do things like look at photographs or read sentences or hear sounds, and we can track using the MRI scanner, where blood is flowing to in their brain. And we know that that's important because when neurons are active, they use up energy, and they require energy in the form of oxygen and glucose to flow to them in the blood. Okay, so back to structural MRI. So in the last 10 or 15 years, various groups around the world have started to track changes in the structure of the living human brain at all ages. And I'll show you a couple of examples of this. So these data are, this is a video, which I'll explain, and I'll show you a few times. This is a video of a large amount of data collected by Christian Tamnes at Oslo. He has scanned hundreds of individuals as they grow up. So these data come from individuals between the ages of 8 and 20, and everyone was scanned every couple of years as they get older. And so what you're going to see is how grey matter changes between the ages of 8 and 20 in the living human brain. I'm going to show you two different images in this video. The first is this one that you can see here. So this shows the outside of the brain, the lateral surface. So this is the left side of the brain, that's the front, that's the back. This is the right side of the brain, that's the front, that's the back, and it's the outside surface of the brain. And as the image goes from yellow to red, that shows a change, a decrease in grey matter volume. And the second image will show you the midline sliced down the middle of the brain, where you can see more or less the same kind of thing. So I'll just play this video a couple of times. The first thing to notice as you go from 8 to 20 is that a lot changes. This is the midline of the brain, right down the middle. So a lot changes throughout this age range. However, it doesn't change in a uniform way. Not every region of the brain is changing at the same time in the same way. It's very region specific. And the general finding here is that changes at the back of the brain happen before changes at the front of the brain. And that's true on the surface and also at the midline. So the back of the brain changes before the front of the brain. The back of the brain contains areas like the visual cortex, whereas the front of the brain contains areas like the anterior temporal cortex and the prefrontal cortex. Okay, so you can zoom in and plot what grey matter does in various different regions. And so I'm going to show you that in the next slide. These are actually data from a different group. These are data from Jay Gede's group at the National Institute of Health in Bethesda. So Jay Gede is one of the pioneers of scanning the developing human brain. And these are one of his, this is a graph from one of his first sets of data. This was on a couple of hundred individuals aged between 4 and 22, scanned every couple of years as they get older. And what you see on this vertical axis is grey matter volume, plotted against age and years from 4 to 22. And what you can see is that for both boys in blue and girls in red, grey matter volume gradually increases during childhood. It peaks at some point in late childhood or early adolescence. That's what the arrows point to. The arrows point to the peak in grey matter volume. This happens a couple of years earlier in girls relative to boys, possibly because girls go through puberty a couple of years earlier than boys do. And then what happens after that is that there's a very significant decline in grey matter volume. So there's a very significant decrease in the volume of grey matter in prefrontal cortex and other cortical regions during the period of adolescence. And that decrease, so this cuts off at 22 because that's the age, that's the maximum age of the people in this study. But in fact, this decline in grey matter volume in prefrontal cortex continues right throughout the 20s and even into the 30s. So when I say decline, some of you are probably thinking that sounds really negative, that sounds like a kind of degenerative process. But actually there's reason to believe that this decline in grey matter volume represents a really important neurodevelopmental process. So we don't know what it corresponds to precisely because, as I mentioned earlier, MRI doesn't give us the resolution to see the brain at a cellular or synaptic level. So we can only really guess what's going on at a cellular or synaptic level, but we think from other data from animal studies and from post-mortem human brain tissue studies that this decline in grey matter volume, at least in part, corresponds to a really important developmental process called synaptic pruning. So synaptic pruning is the process whereby excess connections between brain cells are pruned away or eliminated. The way they're pruned away is according to the environment, to the environmental input. So synapses that aren't being used in a particular environment are pruned away, and synapses that are being used remain and are strengthened. And in that way, this pruning process effectively fine-tunes brain tissue according to the environmental input. It's a really important developmental process, and there's evidence that it's happening during adolescence in humans in prefrontal cortex. It's not the only process that might contribute to this decrease in grey matter, but it's one of the processes that we think might play a role. So what is prefrontal cortex? Well, prefrontal cortex is the part of the brain right at the front, just behind your forehead. It's proportionally bigger in humans than in arguably any other species. Its cellular layout is different in humans than in any other species, even our closest relatives. And it plays a role in all these really important high-level cognitive processes. So prefrontal cortex, we know this from many studies, both neuroimaging studies, but also studies of patients who've had damage to their prefrontal cortex. We know is involved in processes like self-control, controlling yourself, stopping yourself doing something silly or saying something inappropriate. Planning, so planning what you're going to do later on this evening or next week or even in 10 years' time. If you don't have a functioning prefrontal cortex, then planning is very, very difficult, if not impossible. Inhibiting inappropriate actions, so that's things like stopping yourself taking a big risk. Problem solving, multitasking, decision making, self-awareness and social interaction. So all of these processes and many more are processes that heavily rely on prefrontal cortex. And this is a region that is continuing to develop throughout adolescence. So I'm particularly interested in my group in this role of prefrontal cortex, social interaction and understanding other people, and the social brain. So to illustrate what I mean by the social brain, I often show this nice photograph of Liverpool Football Club. So what this shows is two aspects of how your social brains work. Okay, so this is Michael Owen, he's just missed a goal. And the first aspect of your social brains that this picture really nicely illustrates is how automatic and instinctive social emotional responses are. So within a split second of Michael Owen missing this goal, everyone is doing the same thing with their face and the same thing with their arms. Even Michael Owen is as he slides along the ground. The only people who aren't are these guys up here in yellow, but I think that they are on the wrong end of the stadium. And they are doing a different social emotional gesture that we all instantly recognize. And that's the second aspect of the social brain that this picture really nicely illustrates, how good we are at reading other people's behaviour and actions in terms of their underlying hidden mental states and emotions. You don't have to ask any of these people. Just by looking at them, you have a really good idea of what they're thinking and feeling at that precise moment in time. And that ability to read other people's behaviour in terms of underlying mental states and emotions is sometimes called theory of mind or mentalizing. It's a very, very big topic in developmental psychology in particular. When does theory of mind develop? And there have been about 30 years of research on that. Mostly looking at the development of theory of mind in preschool children. And what this research has shown, I'm not going to go into any detail, but just to give you a little kind of snippet, what this research has shown is that different aspects of theory of mind develop at different stages over the first 4 or 5 years of life. You can see examples of theory of behaviour that relies on theory of mind at very young ages, for example, at 18 months. What this video shows of an example of some very nice studies done by Vamaken and Tomasello, where they invited toddlers with their parents into the lab. They didn't give them any instructions, and the rest will become clear when you watch this video. And look at the eye contact at the end. Like duh. That behaviour, that spontaneous helping behaviour that toddler showed relied on an inference about the adult's intentions. That toddler must have figured out, well, that adult intends to open the cupboard and put the books in, but can't because his hands are full, and that's why he helped him. That's the argument. So that's what I mean by theory of mind and how it's used in everyday life. The inference about other people's mental states, like in this case intentions or beliefs, and also emotions. So we know quite a lot about how the brain enables us to attribute mental states and emotions to other people and to ourselves. There's been a lot of research now, probably about 15 or so years of research on on using theory of mind tasks in brain scanners, and what they tend to show is that a very specific network of brain regions is used whenever you think about other people's minds. And this network includes these 4 regions. So medial prefrontal cortex, that's right in the middle of the brain at the front. So this is the front of the brain. And this is the front of the brain, but they're viewed from the side. And these 3 regions in the temporal cortex. So anterior temporal cortex, and then 2 regions right at the back of the temporal cortex, posterior superior temporal sulcus and the temporoparietal junction. These regions are consistently activated in neuroimaging studies when adults lie inside the scanner and think about other people's mental states. So what we're really interested in my lab is how does this social brain network develop in adolescence? And we look at that in various ways. We look at the development of the structure of these brain regions, and also how they function, and also behaviour, social cognitive behaviour. So I'll just give you a few examples of what we've learned. Okay, so one study that we did looked at structural development of the social brain in adolescence. This was a study carried out by Kate Mills in collaboration with our colleague, Jay Geed at NIH. So we used data from his paediatric neuroimaging project. So in this particular study, we included 288 participants, just under half of whom were female, aged between about 7 and 30 years. Each participant was scanned between 2 and 7 times, at least 2 years apart. So this is a kind of data set where we're combining longitudinal data where the same individual is scanned as they get older, and cross-sectional data from people of different ages. So the total number of scans that we had for this study are 857, and what we're really interested in is how does grey matter volume, how does the structure of the cortex change in social brain areas that I've just told you about across this period of life between the ages of 7 and 30. So if I just show you some of the data that we collected from this, so what this shows is grey matter volume in the social brain network in each of these 4 regions that I just mentioned. And what you can see is that in each region there is a very significant and fairly substantial decrease in grey matter volume in each of these 4 social brain regions. And again, we don't know what that decrease in grey matter volume corresponds to at a cellular or synaptic level, but we can only guess that at least in part it might be partly due to pruning of excess synapses. Okay, we can also look at how the functioning of the social brain changes across adolescence. And we've done quite a few studies on this, and to illustrate this kind of work, I'm going to talk about just one study. And in this study, we were interested in the dissociation between puberty and age on social brain development. So what I mean by this is that in early adolescence, puberty stage and age are not tightly correlated. In other words, if you take a group of 12-year-old girls, you might have some who are pre-puberty, who are very early in puberty, and you might have some in exactly the same class, with the same date of birth even, who are quite late in puberty. And that's perfectly normal. There's a very large variation of puberty onset and puberty stage in early adolescence. The question is, what does brain development track? Does brain development track age, chronological age, and how long you've been alive, or is it more associated with physical maturity and puberty hormones? So that's one of the questions that we're interested in my lab, and one of the studies we've done on this was carried out by Stephanie Burnett, a former PhD student of mine who's now in Oxford, and Annalisa Goddings, who's a paediatrician who's doing her PhD with me and Russell Viner on the effect of puberty on adolescent brain development. And in this particular experiment, we were interested in the social emotion processing and how that changes across puberty. So what I mean by social emotions are emotions that require a representation of someone else's mental state. So things like guilt and embarrassment. To feel guilty or to feel embarrassed, you need to think about how someone else would feel about you or think of you think about you as a consequence of your action. So in a way, these emotions are kind of mentalizing emotions, whereas basic emotions like disgust and fear, you can feel them without the need to think about how anyone else thinks about you. So in this particular experiment, we had people lie inside the scanner and read scenarios that were designed to pertain to these different emotions. In this case, there are a few examples of guilt scenarios, things like you laughed when your friend told you she was feeling upset. How guilty do you feel? So they read many, many different little stories that pertained either to guilt or embarrassment or disgust or fear. And we were really interested in brain activity when you compare social emotions and basic emotions. And in this particular experiment, we were interested in the dissociation between chronological age and puberty and their effects on this network of regions. So we scanned 42 girls aged between 11 and 13 and a half. This age range was chosen because that's when the variation of puberty is really maximal for girls. And we took hormonal assays. So we took measurements of hormones from the saliva of sex hormones that are known to increase substantially at puberty. So estradiol, testosterone, and DHEA. So we know that when you compare social emotions and basic emotions, you find activity in this social brain network. The question is, how do different regions of the network change across puberty with the puberty hormones and across age? So one of the regions, this is the left anterior temporal cortex, so that's just behind your ear on the left. Its activity during social emotion processing seems to be associated very significantly with hormone level for all three sex hormones, independent of age. So what this means is that this region's activity, when you're thinking about social emotions, seems to be driven by your puberty hormone levels and not how long you've been alive. And that is exactly the opposite of what we found for a different region of the social brain network. This is the medial prefrontal cortex, a different, very important part of the social brain network. Its activity was associated with age and not with hormone level. So here, this is activity in this medial prefrontal region plotted against age from 11 to 13 and a half, and that's a significant negative correlation. So in other words, as you get older, this region's activity level decreases, and that is independent of hormone level. So what that suggests is that for the medial prefrontal cortex, its activity is driven by how long you've been alive and possibly how many years of experience you've had, so a particularly social experience. Okay, that result has been replicated several times. So this is a meta-analysis that I've been adding to over the years on the different studies partly from my lab and partly from other labs around the world, looking at social brain activity across adolescence, and they all show the same thing. They all show that activity in the medial prefrontal cortex, when people are thinking about other people's mental states or emotions, decreases during the adolescent period. And one of the questions we're looking at now is why this is, and one of the answers might be that the cognitive strategy that people use to do these kinds of tasks changes across this age range. And one of the ways we look at cognitive strategy is to ask whether social cognitive behaviour changes across adolescence. So this is a series of studies that my former postdoc, Iroise Dumontet, who now has her own lab at Birkbeck here in London, did a couple of years ago. So in this particular experiment, we found this task which involves using theory of mind, involves understanding other people's minds and taking another person's perspective, but results in quite a large number of errors even in adults. You need errors in performance in order to see any kind of developmental trajectory. So in this task, you see a set of shelves, and there are various objects on the shelves, and there's a director standing behind the set of shelves. He can't see some of the objects you can see because they're occluded from his point of view by an opaque piece of wood. So this is the same set of shelves from his point of view, and he's never going to ask you to move objects that he can't see. So your task in this experiment is to move objects that he directs you to move, but remembering that he can't see some of them. So he might say, move the top truck up. Now there are 3 trucks here, and the top one from your perspective is this one. However, he can't see this one, so the top truck from his perspective must be this one, and that would be the correct one to move. We know from studies in adults that adults make errors on about 50%, half of these trials. We also introduced a no director condition in which in this case the director's gone and you're given a simple rule to remember. The rule is ignore objects with a grey background. So if an object has a grey background like this one, you ignore it. It's effectively exactly the same kind of task. If you're asked to move objects around, the top truck again would be this one, but you'd ignore it, so you'd move to that one, and again this one would be the correct answer. So just to show you the data on this, these are errors, percentage errors, so the higher the bars, the worse you're doing, in 5 different age groups. These are data from about 180 individuals, aged between 7 and 27. So if you look at the data from the adults here, the blue bars are errors in the director condition and the green bars are errors in the no director condition. You can see that our adults are making errors almost 50% of the time in the director condition, but not in the no director condition. They're much better at that. Developmentally, these 2 conditions have a very similar developmental trajectory, gradually improving between mid-childhood and mid-adolescence, but it's what happens after that that's really interesting. Here, there's no continued improvement in the no director condition between mid-adolescence and adulthood, but there is continued improvement in the director condition. And what that means, we think, is that the ability to take someone else's perspective in order to guide ongoing behaviour in this kind of communication context is still developing at this relatively late stage in life. Okay, so just to summarise the studies I've told you about. The social brain develops both structurally and functionally during adolescence. Social cognitive behaviour changes during adolescence, and we think that peer acceptance is a major influence on adolescent typical behaviour. So adolescence is a really important time of opportunity for learning, for creativity, and for development of self-identity. One of the things, so I've talked about, developing things like how other people see you, what kind of person you are, your political moral beliefs. But another thing that develops often during adolescence are your aspirations for the future. In fact, in this country, we enforce that. We force teenagers to make choices about what subjects they're going to take up at GCSE and A-levels. We force them to think about what they're going to do for the rest of their lives. Far too early, in my view, and far too selective early. However, we do it so they have to think about what they're going to do as their jobs. Some teenagers, like Rosalind Franklin, seem to know very clearly what they want to do when they grow up as a teenager. So this is a quote from the very brilliant biography of Rosalind Franklin by Brenda Maddox. This is a letter written to her father in 1940, so when she was about 20, but she was very clearly wedded to science at school, choosing all science subjects. This is when she was at Cambridge studying natural sciences. Her father wasn't that happy with her choice of career. I think there's evidence that he wasn't that happy that, from a kind of skeptical point of view, that his daughter was becoming a scientist. So she says, you look at science as some sort of demoralizing invention of man, something apart from real life and which must be cautiously guarded and kept separate from everyday existence. But science and everyday life cannot and should not be separated. Science, for me, gives a partial explanation of life. So I wanted to tell you in the last minute or two of this talk about the Rosalind Franklin project that I plan to carry out. When I go into schools to give talks to teenagers or in my work with teenagers, one thing that I'm always surprised by is that the stereotype that scientists are men still very much prevails. It's really shocking to me because actually lots and lots of scientists are women these days. But when you look at the kind of information that teenagers are exposed to about scientists, maybe it's not that surprising. If you put into Wikipedia now the word scientist, what you'll find is this image. This image is lovely and beautiful, but it doesn't give you a very diverse representation of scientists. It's actually not very representative of what scientists look like today. So last year's Rosalind Franklin project was by Professor Polly Arnold. She's created a book and a CD called Chemical Imbalance. And actually, you can pick up this book and CD just outside this room after this lecture if you're interested. And this year's, my Rosalind Franklin project I plan to create a blog, so an online website about scientists' lives. And this will be aimed at secondary school students, so aimed at teenagers, and I will enlist teenagers to help me create this blog. So come up with questions that they're really interested in hearing about in terms of scientists' lives. So I will be interviewing both male and female scientists in equal number across the science subjects, and I hope that it will give them a better representation, a more diverse representation of scientists today. So that just leaves me to say thank you for listening, thank you to my funding and all the people in my lab who've worked on the studies that I've told you about today. Thank you.
Well, thank you very much. That was wonderful. You can tell from the level of applause how much people appreciated it. We now have a little time for questions. There are two people who have microphones out here. So if you have a question, can you please raise your hand and wait till first of all, I identify you, and then that someone brings you a microphone. And please stand up and speak clearly so that it will be on the recording too. So, questions? Yes, lady there with red.
I was wondering if you could say a little bit about some of the differences in teenagers. Just from my own experience, some teenagers care a lot more about what their peers think than others. And understand there's going to be social factors like if you've got, I don't know, might be wrong, but if you've got a more unstable home life, possibly you'll think more about your peers and try and sort of connect more with them. I don't know if that's true, but I was wondering if you could just say a little bit about maybe some of the other factors that affect the massive differences we see in teenagers and their behaviour.
This is an excellent question. So what I've been talking about so far are averages. Most of cognitive neuroscience is about averaging data, and we see individual differences as variance. We try to minimize that as much as possible. But actually, what is probably much more important, especially in terms of adolescent brain development, are these individual differences because they are so vast. So this is a very young field. People have only really been looking at the teenage brain for 10 or so years. They started with averages, saying, what does an average teenager's brain look like compared with an average adult? Only very recently are people starting to look at individual differences. So, for example, how do your genes affect your brain development? How does your environment affect brain development? How does culture affect it? I mentioned right at the beginning that there are huge cultural differences in the expectations of teenagers. How does that affect their brain development? Do their brains undergo this very protracted development that I've been talking about today if their culture treats them very differently? We don't know the answer yet because the studies haven't been done. People are just starting to look at that. There are vast individual differences in things like socio-economic status. I think that's what you're talking about. And they are bound to influence, all these environmental factors are bound to influence the way the brain is shaped and develops as you grow up. And people are just starting to ask those kinds of questions. And there are groups around the world who are starting to look at that. And they're starting to come out with really interesting data. But the answer is it will explain, it will to some extent mould and shape the development of the brain.
Okay, there was a question in the front row.
Could you get the microphone to the interpreter? I'm just thinking about deaf people and hearing people and whether their brains are the same and whether deaf and hearing teenagers have been tested to see whether the brain development's any different.
I'm laughing because one of the world's experts on this is in the audience. It's not my area of research at all, but it is the area of research of Mairead McSweeney, who's at UCL, and she's here in the audience. I don't want to misrepresent her work, but she might be willing to talk to you afterwards. But there are differences in deaf and hearing people's brains.
All right, there's a question there in the middle.
I just remember seeing the graph where it said boys and girls with the grey matter. Is it of any significance that the boys had higher amount of grey matter than the females?
Yeah, so males have more grey matter than females, actually at all ages in their brains. And we don't really know why. We think that a lot of the difference is probably explained by difference in overall body mass. When you take that into account, that gets rid of some of the difference, but actually there's still a difference left even after you've covaried out differences in height and weight. And we don't know why that difference is there, but it's true at all ages and in all brain regions as far as we've been able to see.
There's one right at the back there, gentlemen.
One of the things I've observed is that death rates amongst adolescents going up to the ages of about 25, amongst males is per 1,000 is roughly twice that of females. This presumably reflects risk in some way. Why is there such an enormous difference in risk taking, if that's the cause of this difference between males and females?
This is a really excellent question. So one of the things to say, first of all, is that if you look in the lab, it's very difficult to find significant gender differences in the kinds of things that we measure. That's probably because there are gender differences, but there is also a huge overlap between the genders. So in order to see any difference, you need enormous samples, which we don't have. In everyday life, you're right that there are the leading cause of death in adolescence in most studies is accidents resulting from risk-taking, and there is a gender bias towards boys, towards males. One thing that I would say about that is that the risks, so I mentioned right at the beginning that adolescents take some risks but not others, and actually risk-taking, if you kind of parse it down into various components, is really quite complex. And adolescent girls probably do take risks like adolescent boys do, but different kinds of risks. So for example, taking up smoking is more common in adolescent girls than boys. Teenage girls are more likely to take up smoking than boys. So this is not a risk that is going to end in their death right there and then. So they won't make any kind of statistic in these kinds of epidemiological studies, but it's still a risk nevertheless. So I think one thing to think about is not gender differences in overall risk-taking, but gender differences in the type of risk that teenagers are taking. And there are probably really good evolutionary reasons to explain that, but I don't know anything about evolutionary biology, so I'm never going to guess them. One of the other things that I wanted to say about risk, I'm glad you asked your question because it gives me a reason to talk about this very briefly, because I didn't during my talk, is that often risk-taking is a very good thing. We need to take risks. We need to take risks in our everyday lives. Where would we be without risk-taking? We need to take career risks, education risks. So, for example, I work with lots of teachers, and one of the things that they say often is that their kids in their classes don't take enough risks. So risks like putting your hand up and asking a question, or risks like writing down an answer to an essay question that you're not completely sure of, that you're going to have a guess at or make an argument that you haven't read in a textbook. These are risks that actually teachers often worry that adolescents aren't given the confidence to take enough of. So risk-taking does happen in some situations, but not in others. And what we really need to do is harness this tendency to take risks in some situations in adolescence and apply it to situations where there are real benefits of taking risks.
This gentleman there.
Do you think that the growth in online social networks and things is actually just intensifying the peer pressure element of things, or is there something fundamentally different about sort of online social networks?
Again, this is a very, very good question, and one that we don't really know the answer to. One of the ways to think about this is, how does spending so much time on Facebook and Twitter and say video gaming, how does it affect the developing brain? It probably does affect the developing brain, because we know that the developing brain is affected by the environment and experience and what you do. But we don't really know how. It will affect it, but we don't know how. We don't know whether it's in a negative way or a positive way. Each generation worries about new technologies and the effect of new technologies on the current young population. This was true for the printing press and books, it was true for television and radio and DVDs, and now it's the internet, and we really worry about this. The fact is, though, that we just don't know. The research hasn't been done. People are starting to look at how spending a lot of time on Facebook or gaming influences the brain and brain development, but we don't really have any answers yet. In terms of the quality, like whether it's qualitatively similar or different from everyday social interaction, I mean, it certainly feels very different, but one of the things that you've got to remember is that in all the experiments that we do, almost always, when we're looking at social interaction, it's an online version of social interaction. So when we do experiments in the scanner that I've been talking about, and we get people to think about other people's mental states or emotions, they're not people in the room with the person inside the scanner. They're people represented on a computer screen. So actually, what we know about the social brain probably applies more to online social networking than real life, everyday interaction.
Okay, it's a question in the front.
I don't want to make stereotypes, but I wonder whether things like girls being more emotional and more feeling about things is actually backed up by brain activity?
Actually, my answer would be the same as the previous question about gender differences. It's a really good question, but in terms of data, the field of gender differences is quite a big mess, and I think partly it's because, so some studies find evidence of gender differences and other studies don't, and I think that's partly because, again, there's a huge overlap between the genders, and in order to find any difference, you have to have such big samples. And as you've probably noticed from this talk, the samples that we study tend to be very, very small, between, you know, tens or possibly hundreds of participants, which probably just isn't enough, isn't sufficient to see gender differences.
Okay, well, it looks as if people have noticed it's 7:30 and it's time to draw it to a close. So thank you very much. That's been absolutely wonderful. And now, if you step back, you get the prize. Thank you. Thank you.